is Marco Gobbi, and I work in Milan, Italy at the Mario Negri Institute for Pharmacological Research, a non-profit foundation devoted to biomedical research. I've been doing a label-free analysis for 8 years, using the Protein instrument in the last 5 years. Using Protein, we've obtained crucial data in different research projects, in particular studying the amyloidogenic proteins involved in neurodegenerative diseases, such as Alzheimer and prion diseases. Please listen to the following webinar, where I will describe the quite unconventional applications of the protein system in this research field.
So let me start with a few slides for the introduction of this topic. As you probably know, there are many an increasing list of human diseases which are associated with the aggregation of misfolded proteins, which eventually lead to the formation of amyloid peptides, amyloid deposits. You can see here some of these diseases, and these include neurodegenerative diseases such as Alzheimer's disease, prion diseases, Parkinson and Huntington disease, but also some important peripheral amyloidosis. You can see here that each of these diseases are due to the aggregation of different proteins or peptides. Although the proteins are different, the amyloid deposits they form have very similar features. In particular, the amyloid-like deposits are characterized by very similar tinctorial properties. They are very stable, are highly resistant to proteolytic digestion, and when analyzed by electron microscopy, they show a fibrillar structure. Also, the mechanism leading to the formation of amyloid-like deposits is very similar for all these proteins and involve a first misfolding of the protein, which is followed by a conformational rearrangement, in particular with the formation of beta-sheet structures. These beta-sheet structures increase the propensity of the protein to aggregate and to form ordered structures, which at the beginning are known as protofibrils and then became fibrils. Today I will focus my attention and I will show you some example obtained with amyloid beta peptide, which is the peptide involved in Alzheimer's disease. However, the same concepts I will show you can be also applied to the other aggregating proteins. So, amyloid beta peptide. Amyloid beta peptide is a 40-42 amino acid peptide, which is formed by sequential cleavage of the amyloid precursor protein, APP, and which result in the release of a beta in form of monomers. A beta peptides are very prone to aggregate and to form the fibrils. You can see here some atomic form microscopy image of these fibrils. Fibrils eventually form the amyloid plaques, which represent a hallmark of Alzheimer's disease, and which at the beginning were thought to be the main neuropathogenic species responsible for the dementia. However, more recent studies suggest very clearly that also small soluble intermediate oligomeric species, which form in the pathway leading to the amyloid plaques, have important neurotoxic properties, and actually it is thought that these are the main responsible for the neurotoxicity occurring in Alzheimer's disease. So today I will show you some example on how SPR can be used to study either the elongation of fibrils and the formation of oligomers, which are two very important processes occurring in the aggregation mechanisms. So let's start with just a point as regards fibril elongation. I will show you some data we have published last year. And as regards oligomer formation, I will show you some new data which have not yet been published, but we hope that they will be published very soon. Let's start with the fibril formation and fibril elongation. In general, it is thought that the kinetics of fibril formation is very similar to a crystallization process, with an initial lag phase, which is important, which is necessary for the formation of the nucleus, in particular a beta sheet enriched nucleus, and this lag phase is followed. Lag phase can be very, very long, but once formed the nucleus, there is a very rapid elongation phase and fibril growth until a plateau. The mechanism by which this elongation is thought to occur is that the monomers bind very avidly to the fibril ends, and once bound to the fibril ends, they undergo conformational changes so that they acquire the beta sheet structure, for example, here, and they become a new seed, a new binding site for another monomer, so sustaining the fibril elongation. There are different chemical and chemical-physical approaches to study the kinetics of fibril elongation. Two of them, very used, are circular dichroism, which sees the secondary structures of proteins, and in particular the beta sheet structure, and the use of dyes such as thioflavin T, which are specific for the presence of beta sheet. You can recognize the lag phase, which is followed by a very rapid increase of the signal here, due to the presence of beta sheet containing structure. However, I will show you how SPR can represent a very useful complementary technique to understand details which cannot be appreciated with these techniques. In particular, because the time scale is different. With these techniques, the time scale is in the hours, whereas with SPR, the time scale is seconds. And this is very likely the time scale involved in this binding reaction and conformational changes occurring in the fibrils. So, I've prepared an animation to show the principle of these SPR studies. For these studies, we mobilized fibrils on the sensor chip. Here are the fibrils, and inject monomers in the fluidic channels. The monomers binds to the end, is expected to bind to the end of fibrils, and the binding is expected to be followed by a conformational change, resulting in the formation of a new binding site for another monomer molecule. So, in the association phase, we can expect to look at the elongation of fibrils, which is measured as an increase of mass of the fibrils immobilized on the chip. The dissociation phase is also very interesting because it can be predicted that the monomers which bound at the later stage and which had not the time to undergo the conformational change, can dissociate very rapidly for the fibrils, whereas the species, the monomers, which at the time to undergo the conformational change remain attached to the fibril. I told you that fibrils are very stable structures. So we expect that we can see at the end of the SPR session an increase of mass on the chip due to the elongation of the fibril. This approach actually had been developed and characterized in 2000 by David Michka and co-workers. And they used this approach to study the elongation of a beta-140 peptide. More recently, we have used the same approach to study the elongation of prion proteins, this paper in 2006, and very recently we have also published a paper using the same approach to study the elongation kinetics of the highly amyloidogenic beta-142. The beta-142 is much more amyloidogenic and aggregating than beta-140. It's very difficult to study, and we succeed in doing these studies just last year. How did we perform the experiment? This is very simple. On the proton apparatus, we immobilize fibrils in one lane of the sensor chip, and then after rotation, we inject a beta-142 modules to check for the elongation. This is the core of the experiment. However, we also did some other studies to characterize the approach. In particular, we mobilized also a beta-142 monomers as a control, and also a beta-142 in parallel lanes, and also a beta-142 sonicated fibrils, in order to allow a greater number of fibril ends to be suitable for binding. Okay, and then we inject different analytes for characterization purposes. So these are the main results. We immobilize in 3 different lanes, as I told you before, a beta-142 monomers, a beta-142 fibrils, and a beta-142 sonicated fibrils. The total levels of A beta, we are using here synthetic A beta peptide. The amount of peptide immobilized in the 3 channels was very similar, about 4,000 are used. So the first injection was with an anti-beta antibody. This is a classical anti-beta antibody, 6010, and you can see that the antibody, the antibody resulted in very similar sensorgram in all the lanes. And this was expected since the antibody recognized A beta independently on the structure. And the amount of binding was proportional to the amount of A beta immobilized onto the chip. The second injection was with Congo-Red. Congo-Red is a dye which specifically recognizes beta sheet structures. You can see that, as expected, Congo-Red did not bind to a beta-142 monomers because they do not contain beta sheet structures, whereas Congo-Red bound in the lanes where we mobilized fibrils and sonicated fibrils. There was no difference in the amount of binding in the 2 lanes, and this was also expected since Congo-Red bind to the beta sheet datasheet structures. Okay, after this characterization injection, we did the core, the experiment, the main experiment. And so we injected a beta-142 monomers. So you can see here the main results. The first point is that monomers bind the fibrils, but not monomers. And this can be expected because indicated that fibrils, but not monomers, act as an efficient binding site and likely an efficient site for the elongation. The second point to be observed is that the binding on sonicated fibrils is much higher than the binding on a beta fibrils. This suggests that the monomers bind to the end of the fibrils because there are much more ends in the sonicated fibrils when we mobilize sonicated fibrils than when we mobilize longer fibrils. Perhaps I forgot to tell, when fibrils are a long structure, the sonication of fibrils results in a fragmentation of the fibrils, so that the amount of a beta is very similar, but in this situation the fibril is longer. In this situation, each fibril is fragmented in different fragments. And it was interesting that monomers bind much more in this plane than in this one, confirming the fact that we are looking at the binding to the ends of the fibrils. There is also another important point. The gray and the black sensograms indicate the sensograms obtained in 2 consecutive injections done without any regeneration. The fact that this consecutive injection produced very similar sensograms suggests that the binding does not saturate. And this again is consistent with the fact that we are looking at a fibril elongation and not simply at a binding reaction. When looking at the dissociation, we observed a multiphasic dissociation phase, and also we observed a residual pseudo-reversible binding. And again, this was consistent with the fact that we are looking at the formation of the stable fibrils. All these sensograms could not be fitted by a simple Langmuir equation, but they require a more complex models, and we found that we could fit very well the sensograms by using the conformational change equation, which was chosen because it is the equation which model the dock and lock mechanism, in which the first binding step, the docking step, is followed by conformational changes, which results in a change in the affinity of the species on the immobilized fibril. Okay, with this analysis, we could at the point estimate the kinetic rate constants for a beta-142 elongation. And these are the values, and we could compare these values with those obtained with a beta-140, and we could compare these values with those with the beta-mutated peptides or prion proteins and so on. However, the main reason for setting up this approach for us is to study inhibitors of fibril elongation. This is I work in a pharmacological research institute, and we are mainly looking at compounds interfering or possibly interfering with the diseases. And the possibility to interfere with fibril elongation is one of the manner by which researchers are trying to interfere with beta aggregation. I will show you two examples for these studies. This is the first study we did with this peptide, which was developed by Dr. Olson in England. This peptide was designed is a retroinverso peptide, which was designed to bind a central region of the A-beta sequence. This central region is important for the stacking occurring during the fibril formation, and is important for the fibril formation. So the idea here was that this peptide could act as a sort of beta breaker. This should bind this region, but impair the elongation of fibrils. So we used SPR to test this hypothesis, and this is the protocol we used. As I said above, we mobilized a beta fibrils on the chip, and then we injected a beta monomers. A beta monomers alone or in the presence of the peptide to see if the peptide interferes with the elongation. We also injected the small peptide alone in order to see its binding properties. The results are shown here, and you can see here the red line show the sensorgram obtained when injecting a beta 142 alone. You can see a linear phase without any saturation, and the dissociation was very small, and a lot of binding remained at the end of the session, as expected. The binding of the small peptide is very different. You can see that the small peptide bound to the fibrils very rapidly to a plateau, and then at the end of the injection it dissociated very rapidly. When the two peptides, the beta-142 and the small peptide, were injected together, you can see this is the sensorgram, the blue line, and you can see that at the end of the injection, the amount of A-beta remaining on the chip is much lower than the one obtained when injected A-beta alone, indicating that the small peptide is actually interfering with the elongation. This is much more clear after normalization of this data, that is by subtracting this signal, the signal of the peptide alone, from the signal of the peptide plus A-beta. This allowed to obtain these two sensograms. This is A-beta alone, this is A-beta 142 in the presence of the small peptide. And you can see that there is a very marked and clear reduction of the signal, indicating that this peptide is actually inhibiting the elongation. These experiments also indicated us a mechanism by which this peptide inhibited elongation. This peptide binds the fibrils much better than the beta-142, so compete with a beta-142 for the fibrils. But however, it cannot sustain the elongation, so its final effect is to impair the binding and the elongation of a beta-142. Very similar results were obtained with another small peptide. This is a beta-1,6. This is a small peptide with a mutation designed on the N-terminal of a beta-140. This peptide was designed according to the data obtained in this paper, published in 2009, where we described this important mutation. However, here I don't have no time to enter into this topic. However, for these studies, in order to check if this small peptide can interfere with the elongation of a beta-140, in this case we used a beta-140, we mobilized fibrils on the chip and injected monomers in the absence of the presence of the small peptide. The results are shown here. The blue line shows the sensorgram obtained with a beta-140 alone. Again, no saturation, very small dissociation. Again, the small peptide bind better, faster than the longer peptide, but reach like a plateau and then dissociated very rapidly. And we injected the two peptide alone, we found a very clear decrease of the final signal. Again, we can normalize this data to show that the presence of a beta-1,6 markedly reduce the growth of a beta-140 fibers. Okay. So after these results obtained with when studying fibril elongation, the next part of the webinar will be dedicated to the SPR studies for looking at the formation of AB oligomers. As I told you before, these are new data, and we hope that they will be published very soon. I remember that AB oligomers are small soluble intermediates occurring during the formation of during the aggregation process, and thought to be and found to be very neurotoxic and thought to be very important pathogenic species. So there is a very great effort of many research groups to try to study better these species, these small species. This is a quite difficult task because these species are transient species. They are very unstable. Their structure is unknown, and also there is a high heterogeneity. It could be that it's like that there are many different oligomeric species present at the same time. And this is a very recent paper published last week on JDC, and showing that there are different oligomeric species with similar size, so they are small oligomers, but showing different toxicity. And the difference between these different oligomeric species is a structural difference, conformational differences. In particular, toxic oligomers were shown to expose hydrophobic residues, whereas non-toxic oligomers do not. So there is a need to find and to develop tools to recognize oligomers, and in particular to recognize the toxic oligomers. This is a major need in Alzheimer's disease. How can these subtle differences in these subtle structural differences be recognized. One of the most suitable approaches employs antibodies. And so I will show antibodies which can be very useful to distinguish between different structures. So I will show you some data obtained characterizing a new surface plasmon resonance-based immunoassay for toxic oligomers. The idea is very simple. We mobilize the antibody on the chip and we flow a beta peptides at different stage of their aggregation process. The hope was that antibodies could recognize in a different manner oligomers from fibrils or monomers. So to differentially recognize oligomeric species, and even better, to recognize the toxic oligomeric species. This was the main aim of our study. Our feeling was that SPR has many different advantages over well-known immunoassays such as ELISA or dot plot. The readout is direct and in real time seconds, whereas with ELISA or dot plot you need long incubation and washing steps. And this is important when studying transient species which can evolve to form other species during these long steps. The second point is that SPR, but not ELISA or dot plot, might allow to distinguish species on the basis of different binding constants. And the possibility to measure and to differentiate according to different binding constants might be a very important advantage over ELISA or dot plot, in which the differences in affinities are probably level out. So we screened the different antibodies, and I will show you the results we obtained with 4G8, which is a very classic anti-A-beta antibody, which recognizes a central domain of beta. It's also important that this is an heterophobic domain of a beta. And then we injected as analyte a synthetic beta-142. We prepared a solution of a beta-142 and took aliquots of the peptide from the same solution at different incubation time points. That means at different points of the aggregation process. This is the sensogram we obtained when injecting the peptide, the freshly prepared peptide, that is at time 0. When we injected the peptide after 2 hours incubation, this is the same solution, but the only difference is the incubation time, you can see that the sensogram is completely different. The signal is higher, and also the shape is different. And this already is indicating that SPR is detecting some changes occurring in the A-beta solution. When injected the peptide after 5 hours incubation, we still observed an increase of signal from 400 to about 600. But if we wait 8 hours and then inject the peptide, we start to see a decrease of binding. And the decrease continued at longer time points, and at 72 hours, the binding signal is very low. So, SPR and this antibody, this immobilized antibody, is sensing some changes occurring in the solution. These changes are either quantitative, you can see that the absolute binding is different, there is an increase, and then a decrease, but also a qualitative difference. The shape of the sensogram is completely different. No one of these sensograms could be fitted by a simple bimolecular Langmuir equation. And we observed that a very nice fitting could be obtained by assuming the presence of two binding species. In particular, two binding species with showing different dissociation rate constants. So each of these sensogram, the black lane is the raw data, could be is made up by the binding of two species. Let's look at this one. One species is characterized by a very fast dissociation rate constant, and the other component is characterized by a very slow dissociation rate constant. You can see that the fast payoff species are the main species present at T0 and decreased here, whereas the slow payoff species are negligible here, increase here, and then decrease again to 0 levels after 72 hours. These results were obtained by a global fitting of all this data. So all this curve, this experimental curve, were made by two components with a fast payoff with this value and the slow payoff of this value. We then try to characterize the Abeta species corresponding to these fast payoff and slow payoff species. We used different chemical-physical and biochemical approaches to do that, but here I show you just studies with size exclusion chromatography, SEC. This is the SEC profile obtained with T0, when we only found the fast payoff species. And in this condition, the SEC profile indicates the presence of monomer peak only. So this data suggested us that the fast dissociating component is actually due to the monomers. And we knew the concentration of monomers there, and we can estimate the affinity of monomers for the antibody. What happened at 5 hours? That is the time points at which we had the maximum level of the slow payoff species. When we injected the solution in SEC, we found two main peaks. And you can see that compared to T0, there is a decrease in the monomer peak and the appearance of a peak in the void volume. So, what was likely is that the species present in the void volume, that means aggregated species, were the same appearing here as the slow payoff species. To validate this hypothesis, we made an experiment in which we injected in SPR the two fraction obtained from SEC. And what we found is that if we inject the fraction corresponding to monomeric peak, we obtained the fast dissociating species as expected. Whereas if we inject the fraction corresponding to the void volume, we obtained the slow dissociating species. And this confirmed our hypothesis, and is telling us that SPR is suitable to recognize oligomeric species in solutions also containing monomeric species. We did experiments, and the same results could not be obtained with classical immunoassays, such as ELISA or dot plot, which were not able to distinguish these two components. I have no time but to show the data now, but we also did a lot of studies showing that let me continue here. After having characterized that the slowly dissociating component is due to the aggregated oligomeric species, we have the tool to dissect the kinetic of appearance and disappearance of the species. And this is a very important finding which could not have been obtained with other immunoassays. We also obtained evidence showing that the oligomeric species detected by 4G8 in this SPR approach are actually the toxic oligomeric species. I have no time, but this is the main point of our paper. So, we have a tool which is able to detect and to recognize toxic oligomeric species in a complex solution. In order to, as I told you before, we are interested not just to study the kinetic of oligomer formation. We also did studies looking at the formation of oligomers, looking at how mutation affect the formation of oligomers, but we are mainly interested in using this approach to check for the effect of inhibitors of the formation of oligomers. And as a proof of principle for that, we used epigallocatechin gallate, which is a compound previously found to shift the aggregation pathway and to impair the formation of toxic oligomers. So we test this compound in our assay. This is the sensogram obtained incubating AB142 for 5 hours in the absence of epigallocatechin gallate with the presence of the two species. When we inject a solution of a beta pre-incubated in the presence of this compound, we found a selective inhibition of the formation of toxic oligomers, whereas monomers were not affected. Another important point was to see if this immunoassay can recognize native oligomers. All the studies shown before were obtained with synthetic beta. For that, we used as a proof of principle lysates from transgenic C. elegans, which produce beta-142 oligomers after induction. And when we injected lysates from these worms, we obtained these sensograms, showing a clear signal. Whereas when we injected lysates from worms not induced, that is, not producing a beta or control worms, we didn't find any signal. And also you can see that the shape of this sensogram is the fingerprint of oligomers with a very slow dissociation rate. So in summary, I've shown you some data demonstrating that SPR can be used to investigate the fibril elongation, to get details on the very fine kinetics, and in particular to check for the effect of inhibitors. And I've shown you two examples for that. Very recent data showing that with an SPR-based immunoassay we can recognize toxic oligomers. We can dissect the kinetics of appearance, disappearance of these oligomers. We can look at the effect of inhibitors, and we can look at the effect of mutations. And we can also use this assay to detect native oligomers in biological samples. And we are now doing experiments to check for these species in human samples. Let me finish with some acknowledgments, in particular Matteo Stravalacci, which is a student in my lab working on SPR since for 4 or 5 years, and he did all these studies. And I also have to thank people from the department who helped us to prepare and to characterize the peptides used for the studies. And I thank you for the attention.